Hello, I'm Ushma Neill with the Journal of Clinical Investigation and the series Conversations with Giants in Medicine. Today I'm thrilled to be joined by physician scientist and gene therapy pioneer Dr. Katherine High. After a long career as an academic hematologist, first at the University of North Carolina, then at the University of Pennsylvania and HHMI, Kathy transitioned to a role in industry, first at Spark Therapeutics, then at AskBio, where she led the first FDA approval for gene therapy for a genetic disease. Thank you so much for joining me today, Kathy.
Happy to be here, Ushma.
So it's a thrill to see you again, and maybe even to learn a little bit more about you. Do you think you could start by telling me a little bit about what you were like as a child?
Well, I was a child who really liked to go to the library, and I read a lot of books. After having had a lot of books read to me, and I really liked math, and some of my early memories are of doing assignments that I had from my dad. For example, writing from 1 to 1,000 when I was about 6 years old. I had made the mistake of thinking that after 100 comes 200 and then 300 and then 400, and so I was rewarded by having to write out all the numbers between 100 and 1,000. But I did learn to do some other things. I learned to play the piano and the viola, and I was on the swim team where one of my sisters was a great champion, so that was a valuable lesson in what it's like to not be at the top of the class. So that's what I was like as a kid.
What did your parents do?
My dad worked in marketing for a very large photographic studio in the Southeast, which actually did the catalogs for Sears, JCI, and Montgomery Ward. So most of his business was in New York and Chicago, but he would not move there. And so we lived in North Carolina. My mother was mostly working at home. She also did some editorial work as we got older. Her father was a chemical engineer, and he headed a large research operation in our city in North Carolina.
So I read in an interview that you've given before that at age 10 you got a chemistry set from Santa Claus,
Yes.
And that began a lifelong love of chemistry.
Yes, my dad, so I was the oldest of 3 daughters, and my dad was determined that he would get a scientist or an engineer out of those 3 children, and I think I seemed the most likely. And so yes, Santa Claus did bring me a chemistry set, and it had all the chemicals you needed to do about 100 different experiments. And it also had a little alcohol lamp so that you could heat reactions and so forth. And so I did a lot of those experiments with my dad. Yes, that was the beginning of my introduction to science.
So then what was the motivation to pursue chemistry as an undergrad? And you clearly were a phenomenal student because you did your AB in chemistry at Harvard University.
Beginning when I was in the 9th grade, my dad took me every year to interview at MIT. And he only had one goal, that I would decide to go to MIT. And as you indicated, I did not decide that. Why was I interested in chemistry? I don't know, maybe based on that chemistry set that I got at age 10. I found it more interesting than physics or biology, the other science courses that I took in high school. So despite a lot of pressure from my dad, who was hoping I would go into aeronautical engineering and then go work for NASA, I stuck with chemistry.
So then when you were at college, was it a love affair with chemistry, or did you start to do?
I really did enjoy majoring in chemistry, and then when I was a senior in college, I thought, well, what if I decide to go to medical school? I should take 1 year of biology, so I did that. And I actually thought that was pretty interesting too. And so in the first year that I got out of college, I took a job working in a pathology lab at MGH. They were a research group. They wanted me to synthesize oxidized derivatives of homocysteine, and then they were injecting those into rabbits to see if they could recapitulate the advanced atherosclerosis that occurs in people with homocystinuria. So they were trying to figure out what was the active metabolite that caused that to happen. So I was happy because I was just doing chemistry, and nonetheless, it served as a good introduction to me about research in medicine. So I thought, well, okay, this looks all right. So I did apply to medical school.
You returned to North Carolina.
I returned to North Carolina.
For
yes.
medical school and stayed for your residency. So
yes.
you did decide to do that. What was the tipping point, and did you think you were going to be a clinician?
I wasn't really sure, and after the first 2.5 years of medical school, I decided to take some time off and return to the chemistry lab. I found a great position in a lab that worked on polymer chemistry, and I was working on the auto-acceleration of the vinyl monomers. And I was remembering then how much fun I had working in the chemistry lab, and I concluded after I'd been there for about 6 months that I should leave medical school and go to graduate school in chemistry, that I had made a mistake. So I went to talk to the head of the lab, and my work there had gone very well, and I had a publication in the Journal of the American Chemical Society, and he told me then, look, once you have a terminal degree, no one cares what it is. And if you really think you'd rather do chemistry, the fastest thing for you to do is to go back, finish medical school. You only have 1.5 years left, and then I'll help you get a postdoc. And if you think that you have deficiencies in your chemistry background, which clearly I would, I hadn't been to graduate school in chemistry, you can take courses while you're a postdoc. So that's my advice to you. And I thought, wow, that makes a lot of sense. So I went back to medical school with the intent to finish up and then go to do as he suggested. But the rotations that I had when I got back were the ones that I turned out to like the most, pediatrics and medicine. And so then I shifted my career goals again and decided that I would finish medical school and I would do training in internal medicine. And I tell you this story just because I think a lot of people in their 20s are uncertain about what they really want to do. And it's just my way of saying, hey, that's okay.
So your residency was in internal medicine.
Yes.
So what turned you towards hematology? Because then you did end up doing a clinical fellowship at Yale in hematology.
Well, I really wanted to go into a subspecialty where I could use my chemistry background. And so I rapidly narrowed it down to endocrinology, hematology, or rheumatology, immunology, because I felt that those were the 3 subspecialties that at that point were best understood at the molecular level, where my chemistry background might be an asset. And so then I tried to think about which patients I liked the most of the ones I had seen on the medicine service, and so that was how I decided for hematology.
Tell us a little bit about that time. So you ended up working in the lab of Ed Benz, who's
Yes.
rather notorious
Yes.
for any number of his accomplishments.
Yes.
But globin genes, what was it that turned you on in terms of the laboratory?
Well, the great thing about what was going on, not only in Ed's lab, but also in Bernie Forget's lab, who was the division chief at that time, was that they were really doing molecular dissection of what causes thalassemia, what are the mutations in the globin gene that are associated with thalassemia, and I found that very exciting and interesting. Now, most of my work moved on to working on the MYC oncogene, actually, which was a project that Ed had undertaken and assigned to me. But during the time I was in that lab, the genes for factor 8 and factor 9 were actually cloned, were isolated and characterized. And so I realized that it would be possible to explore the molecular basis of the hemophilias in exactly the same way that people had already done for thalassemias, enabled by the cloning of those genes. And so that was really the jumping off point for my independent career. And as you know, I took my first faculty job at UNC Chapel Hill. They had a very large research operation in blood coagulation there. And that spanned the gamut from chemists who worked on purifying factor VIII from plasma, from serum, and all the way through to a very large hemophilia center with abundant patient material. And so when I started my independent lab in 1985, our initial projects were several-fold. We were delineating defects in people with hemophilia, and I focused initially on hemophilia B because the gene was smaller and was a little more tractable. It will be surprising to the listeners, I think, that there was no PCR when this started. And so we were making libraries of DNA and then screening the libraries for fragments of factor IX, which we would then sequence. The second project that I was working on was trying to define the defect in the hemophilia B dog colony that they had there. And again, I started by doing low stringency screens of a cDNA library made from canine liver. By the time I had isolated the normal factor IX cDNA, the normal canine factor IX cDNA, the idea of PCR was present. There were no PCR machines, but you could set up 3 different water baths in the lab and move the test tube every 60 seconds or whatever it was, which was still easier than making a library. And so we were able to define the defect in the hemophilia B dog model. And the other thing that we were doing was expressing mutant proteins and then doing structure function studies with colleagues at UNC Chapel Hill. And we began to do experiments with gene transfer just into cells. But part of the motivation for that was that that period, during the late 1980s, was the period when HIV was becoming very, very manifest in the hemophilia population. And as I mentioned before, this was the largest hemophilia clinic on the East Coast. And we knew early on that nearly all the severe hemophilia patients, A and B, were HIV positive, but nobody knew what that meant. And what our friends in infectious disease told us was that, well, most viral diseases are not fatal. Rabies is fatal, but most other viral diseases are not fatal. Well, of course, as we all know, that turned out not to be the case. And we did not have any antiretroviral agents at that time. And after a long time, we only had AZT, which as a single agent, I think, was of limited utility. There were a few patients who responded to a single agent, but most of the time I couldn't tell that we were making much of a difference. And so I was attending on the clinical coagulation service for about 3 months out of the year. And the service rapidly became overwhelming as the number of AIDS patients in the hospital increased. And it became very difficult to manage anything that was going on in the research lab during the months when you were attending, to say nothing of the fact that it was very heartbreaking. These were all young men who had been instructed to fight their disease. They could control it, talking about their hemophilia. That was the attitude that they brought to HIV. And as you know, in those early days, that was not an effective attitude. And so I received a job offer from Penn, and it was going to be in the Department of Pediatrics, and of course I'm not a trained pediatrician. And my clinical responsibility was going to be running the hematology and coagulation laboratories, which is a much more manageable responsibility in terms of you can block out the time for it, and that's the time you're going to work on it, and then except for emergencies, which occasionally occur, you will be able to, in a more consistent way, supervise what's going on in the laboratory. And so to me, that was a very attractive offer, and I ended up accepting it. Now, my colleagues in the Department of Medicine at Penn figured out after a few years where I was, and so I started attending at Hub. but those few years that I was able to alter the balance between teaching, education, teaching, clinical care, and research made a huge difference for what I was doing.
I'm sure. So at Penn, was your clinical footprint significantly smaller?
It certainly was at the beginning, because I was not attending on a busy coagulation service filled with AIDS patients. Actually, when I first came to Penn and CHOP, there were pediatric hemophilia patients there, but no adult hemophilia patients. They all transitioned across town to Jefferson, which had a big hemophilia center. Subsequently, Penn recruited Barbara Kunkel, who set up an adult hemophilia center there. But when I was first there were no adult hemophilia patients. So I was taking care of the teenage patients. I felt comfortable doing that, but not pediatric patients.
So the research evolved over this time as well, from your transition to North Carolina to Penn, as did your patient population. When did you start to fixate on these gene transfers and becoming gene therapy? There were starting to be some initial successes in the literature, but what was it about this world that intrigued you and then you dove headlong into it?
Well, I think for me, one of the major motivations had to do with AIDS in the hemophilia population. And I kept thinking to myself, if only these people had their own gene and could make clotting factor themselves, they wouldn't have to worry about hepatitis C, they wouldn't have to worry about HIV, they wouldn't have to worry about any of these things. So it was while I was at UNC Chapel Hill that I first began to think about this would be important. And it was one of the reasons that we cloned the normal canine factor IX gene, so that we could, the hemophilia dog colonies have been important for the development of almost every therapeutic that we use in hemophilia today. And I thought, okay, it would be great if we could develop vectors that we could use to treat the hemophilia dog model, and then that should easily port over. That's what I thought to myself. And so we had begun that work on gene transfer at UNC Chapel Hill. And I did think, because Bill Kelly was the dean then, and he was aggregating resources around gene therapy, he was recruiting people who were interested in gene therapy, and I thought, all right, well, this could really help our efforts. And so we began the work that we were doing there. We were agnostic about the vector. We made adenoviral vectors, we made AAV vectors, and we really wanted to see what worked the best. And the issue was that adenoviral vectors were triggering a pretty brisk immune response, even in the mice. So we were more interested in moving forward with AAV, and so we were able to show in 1997 that we could cure hemophilia in a mouse using an AAV vector. And in 1999, we published our first paper curing hemophilia in the dog model. These dogs are about 20 kilograms. So in order to do that, we had begun collaborating with a gene therapy company on the West Coast. It was difficult to make enough AAV in those days to treat a 20 kilogram dog. And the only people who were really able to do that were all at companies, gene therapy companies, and they were all on the West Coast. So I talked to all, there were only 3 of them. I talked to all of them, and one of them, which was in San Francisco, Avogen, was interested in the program, and they were spending a lot of their resources on streamlining manufacturing. Mm-hmm. And so we started collaborating. They made the vector to do the experiments in the hemophilic dogs. We collaborated with our former colleagues at UNC Chapel Hill who had the dog colony. We were very excited about the result, and we thought, okay, great, we'll just move this into humans, and And so, as you know, it was a longer story than that. And the first trial that we did, which was the first trial of AAV in skeletal muscle, we could show on a muscle biopsy that we were making factor IX, but we were never able to get adequate circulating levels in an adult male the way that we did in the dogs. But it was safe. So because it was safe, we thought, all right, let's go to the liver, where even in the dog you get better levels. Because, as you know, hepatocytes secrete what they're making directly into the circulation. They don't have to try to make it through all the extracellular matrix in the muscle. And so, sure enough, we did it as a dose escalation study, and when we got to the dose that worked in the dogs, we saw great levels of expression in the first patient who got that dose. And I remember then thinking to myself, wow, I've screwed up our program. I should have been working harder on the preclinical stuff for factor VIII, because now factor IX is done. And of course, as many people know, after about 6 weeks, the levels in that patient started to slowly decline, and they eventually disappeared. And, you know, that was such a difficult experience, even for us in the lab. And for the patient, I cannot imagine. This patient was a physician himself. And he talked about that one day, when it was a very busy day, in the pediatrics clinic, he was a pediatrician, he turned around and slammed his hand on a cabinet when he was trying to make an infusion for a kid. And he thought, oh, I have to treat myself. And then the nurse came and got him and said, oh, you have to see this kid in room 2 right away, bad asthma attack. And so he said the whole afternoon unfolded like that. It was one stress event after the other. And at the end of the day, he looked down at his hand and he hadn't bled. And, you know, so he thought, wow, this is really working. And so then for him to lose it, I mean, it was you know, it was very disappointing for all of us, and especially for him. And, you know, another hemophilia patient described that once as he touched the rainbow.
And then had to come back into the rain.
Yeah, exactly. So we spent the next several years trying to develop reagents that we needed to, so we did hypothesize from the beginning that this was almost certainly an immune response, and that we had not seen it in dogs or mice because they are not natural hosts for AAV, which we are. And that all of these humans probably had some sort of memory response to AAV that was gumming up the works.
Right.
And so we were able to produce evidence for that. We developed reagents like pentamers so that we could directly measure the number of capsid-specific T cells. We actually cloned T cell receptors that were specific for the AAV capsid, and we're able to demonstrate that, you know, the cells, the transduced cells, displayed the cognate peptide MHC complexes. And so we felt like we had a reasonable handle on what was going on. And we proposed then that a short course of immunosuppression would allow the capsids to be degraded and cleared from the cell, and then the patient should get durable expression. You could stop the immunosuppression, patient would have durable expression. And we felt like the importance of what we had learned was that the immune response was not to the transgene product, which is very, very important, because if that were the case, well, that's a non-starter, right? That's a showstopper. You can't pursue this anymore. So the other thing that we learned through all of those molecular studies was one that's not surprising to people with an immunology background. The immune response was dose dependent. And the pathway that was getting used to present this preformed antigen was a very inefficient pathway. So we were not synthesizing the capsids in the cells. We were just taking preformed antigen, breaking it down, and then presenting it. So to the extent that you could lower the dose, you would reduce the risk of that immune response, and you would make it easier to control. And so eventually, we used a naturally occurring variant of factor IX, a high specific activity variant called factor IX Padua that was originally described by one of my colleagues in our CHOP hematology division. It was a kindred from Italy where the proband, who was in his 20s, presented with a deep venous thrombosis. And when the hematologist there worked him up, the only thing that he could find was that the patient had a factor IX level of 770%. So they sequenced his factor IX gene, and he had a single point mutation, and it made a much higher specific activity variant. So we put that into a capsid that we had developed at Children's Hospital that was really hepatotropic, and then we were able to lower the dose of the vector.
No immune response.
And either get no immune response or one that was easily controlled with a short course of steroids. And so that was very exciting, and that was our phase 1/2 trial. Now, by the time that happened, we had already formed Spark. So to backtrack a little bit, in the late 90s and early 2000s, there were some high-profile adverse events in gene therapy. They were not going on in the work that we were doing, and I should have realized that they were likely to affect all of gene therapy, but I was keeping my head down, getting my own work done, and not worrying about the climate around me, and that, of course, was a mistake. But the climate around was becoming very skeptical about gene therapy. And eventually, the company that had been making our vector for the dog studies and then for our early phase clinical testing could not raise money to work on gene therapy anymore, so they turned away from it. And the only way that we were going to be able to keep going is if I could find some other source of clinical grade vector. And I was hoping that Children's Hospital would support me to do it in the hospital.
So I'm very impressed by this phase. You know, gene therapy was considered dead. And no one was going to touch it. And you're being very diplomatic, but some of these high-profile events happened at Penn.
Yes.
And so what kind of power of persuasion you have to have gotten CHOP and Penn to invest in a high-grade like vector development program?
Well, I have to say that to me, one of the most surprising events of my entire career was that when I went to the CEO of Children's Hospital, who is a physician himself, and told him, if you don't help me, if you don't let me set up vector production in the hospital, then I'm going to have to stop everything that I'm doing. I can't go forward because there's nowhere else to get the vector. And when I told him all that, he said, all right, I'm going to have to think about this. And at that point, there were headlines in the Wall Street Journal that said things like, gene therapy cursed or inching toward credibility, you know. And I thought, oh, he's never going to say yes. And so when I walked out of his office, I was trying to think about what's my plan B here? How else can I address this? And I went back to see him a week later, and to my great surprise, his answer was, okay, Dr. High, you keep telling me that there are no problems here that can't be solved. And if that's true, gene therapy will be very important for children's hospitals because we bear the brunt of genetic disease. So I'm going to find the resources for you. I'm going to find space. I'm going to find the money. You're going to hire the people. But I have one request for you. And of course, at that point, I was going to say yes no matter what it was, right? So he said, I don't want you to spend all the money on hemophilia. You have to work on some other diseases that affect the pediatric population. So I said, okay, that's no problem. Because I had long admired Jean Bennett's outstanding data in a dog model of a form of congenital blindness. And I kept trying to get the company in Northern California to work on it. And they invited her out to give a seminar, and the CEO told me, no, it's too small an indication. It wouldn't make sense for us. And I said to him then, what are you spending on public relations? Take that money and spend it on her program. You won't need any money for public relations.
If you can show children with blindness are cured, that will be enough public relations.
But I could never convince them to do it, so I don't have unlimited powers of persuasion. But I did then go to Jean and ask her if she would work with us on a trial that we could do at Children's Hospital, and that we would produce the vector, we would work on getting the clinical trial approved, through all the regulatory hurdles that gene therapy had to go through then, which included not only the FDA, but also the NIH Recombinant DNA Advisory Committee. And so in 2005, we started working on that together. And in 2007, the clinical trial started. And even at the beginning, it seemed pretty clear that a therapeutic effect was occurring. And when you put very small doses of AAV into an immunoprivileged space, like the subretinal space, you don't have to worry about the immune response, was what we saw. I mean, we actually looked for it. We looked for antibodies. We looked for T-cell responses. I mean, it's really, I mean, occasionally you'll see something in the serum, but for the most part, nothing. So we thought that once we had done the dose escalation in the first eye, that we would be able to go to phase 3, but the FDA said, no, you need to take the dose you're going to use in phase 3 and do the contralateral eye in all those same patients to show us that that's safe. So we did that. But by 2012, we were really ready to start the phase 3 study. And we had, I thought, a very collaborative relationship with the Office of Cell and Gene Therapy at the FDA. There were important hurdles in this. There had never been a treatment, a pharmaceutical treatment, for inherited retinal disease. And so we had to develop an endpoint. And we did that very much in dialogue with the FDA. They made many suggestions to make it more rigorous and bulletproof. And so we began this phase 3 trial in 2012. And then the same CEO of the hospital called me in and said, Dr. High, what exactly are you planning to do if this works? Because we're a hospital and we don't commercialize products.
So is this part of the reason why you left Penn to start Spark?
Yes. He formed a subcommittee of the board to look at options. I was getting cold calls in my office from investors, and we were also getting inquiries from large pharma and big biotech that were focused on rare disease. In the end, I was worried about putting this program into a large company that was not focused entirely on gene therapy, because I was afraid that we would encounter some other problem that we had not already solved, and that they wouldn't want to put additional resources into the program for this relatively small indication. So I was really afraid to do that. I thought, we've put so much into this. We've invested so much of our time and our effort and our resources into this, and I don't want to put it in a position where it could be killed. So in the end, we felt, okay, the safest thing to do is to put it into this gene therapy company that we are starting.
But tell me, did you and Jean have a giant bottle of champagne when that first FDA approval came in for Luxturna?
Well, we had to have an advisory committee meeting. And so this is, the FDA doesn't do this for every drug that they approve, but sometimes they assemble a panel of experts and the sponsors have to present all their data and go through an all-day long oral exam on the data and the implications and so forth. And so, of course, we did get assigned for that. We had a presentation that probably had about 100 slides in it. We had 900 backup slides. And I was chairing for the sponsor, and we were up there all day long. And around 3:30 in the afternoon, the chair of the advisory committee said, well, I think we've had adequate discussion now and it's time to vote. And I remember then sitting down and I almost thought I was going to start crying, because I thought, wow, I've been working on this for 12 years and now my fate is going to be determined by something I can't do anything about. I'm just going to have to see what happens on this vote. And it was a unanimous vote in favor of approval. So that was really, really exciting, and I was exhausted, and most of the people went back from Washington to Philadelphia on the bus, and the bus had champagne, but I went back in a car so that I could take calls from reporters.
Well, congratulations. Now,
so I had to wait and have champagne later, Ushma.
But you did get your champagne.
Yes, I meant.
Right. Now, I noticed from your CV that you actually did spend 5 years on an FDA panel,
Yes.
an advisory committee on cell, tissue, and gene therapy.
That's right.
Do you think you learned something from it, or was it endlessly frustrating? Was it helpful? I mean, you've seen the field grow, you've grown it yourself, but this was also an insight into a governmental agency. Any chance you're going to go into politics is the next chapter?
No.
You answered that very quickly.
We had a terrific chair of the committee then. He was a professor at UCSD named Dan Solomon. Wonderful guy, nephrologist, transplant nephrologist by training. And in those days, of course, we were not dealing with product approvals in cell, tissue, and gene therapy. We were mostly dealing with issues about development of regulatory infrastructure. How do you decide that a cell therapy is safe? How do you decide that a gene therapy is safe? What sort of assays do you need to look for recombinant, replication-competent retroviruses, or how much empty capsid can you have contaminating an AAV capsid lot? You know, all those kinds of things. And I think you don't realize how much regulatory infrastructure has to be built in order to allow the regulators to declare a new class of therapeutics safe.
In the other car rides back and forth between all of the facilities at Spark, all of the little minutia that you have to do. So at the end of a long and complicated day, when you think, I should have chosen this as a career, what is that alternative career?
You mean other than science or medicine?
There are other careers out there, Kathy. What else do you think could have kept you entertained?
Well, as I said at the beginning, I always enjoyed reading. I don't know, I guess I would have been a teacher.
Fabulous. Well, I think there's a lot of people with genetic disorders that are being corrected, whether they exist now or a couple years in the future, who are very glad that you took this path. Thank you so much for joining me today, Kathy.
Thank you, Ushma.